Good morning everyone. Today we are going to discuss the topic formation of germ layers and neural crest cells. Okay, so let's start from the embryo. To know about the formation of germ layers, first we need to know that it all starts from after fertilization of egg and womb, which starts the formation of embryo. After a series of differentiation, embryo changes to a structure called as morula. After a series of cell differentiation, morula changes into blastula. Then the blastula changes into gastrula. So here our main important part rotates around this blastula. So this blastula, in this part, the embryo is round in structure with two types of cell masses. Till morula you will have single type of cell structure, whereas blastula have two types of cell structures. Here you have the innermost layer called as trophoblast. So what is this layer called as? Trophoblast, and the remaining cells will condense at one point and it is called as inner cell mass. Okay, so this is the main two layers of the blastula. Now in the next step the blastula will convert into gastrula. This step is called as gastrulation. What happens in the gastrulation? This inner cell mass and trophoblast is present. This inner cell mass is again divided into two cells called as epiblast and hypoblast. So suppose this is the embryo which we have drawn. This is the inner cell mass, which again changes into the outermost layer called as epiblast and innermost layer called as hypoblast. So the epiblast here, this structure is called as amniotic cavity. This structure is called as yolk sac. And again it have epiblast, hypoblast, and then you will have here trophoblast. So this is the structure of the embryo during the phase of gastrula. Now what happens here? From this gastrulation we will discuss about the formation of germ layers. So what are the three germ layers? They are ectoderm, mesoderm and endoderm. Now how these three layers are formed? These three layers are formed from a structure called as primitive streak. What is it called? Primitive streak. Now how this primitive streak is formed? Again come to this structure. What we have seen here? Here we have seen the epiblast and hypoblast, right? These two structures will form primitive streak. These two structures will form what? Primitive streak. So let's come to this figure. Now again I will draw this figure. In this part your embryo will be pear shaped or oval shaped. Okay, and you will have this layer that is called as epiblast, and you will have this layer that is called as hypoblast, and you will have central cavity. So all this is surrounded by the epiblast cells. Now suppose, see, this is embryo. Till half you have epiblast, till half you have hypoblast. Now we will see the embryo from this structure. So from this structure, how you will see your embryo? You will see your embryo like this. Okay, and all this part is your epiblast. Okay, now this is your cranial end and this is your caudal end. What happens here? All the cells of the epiblast, they will invaginate and come in the center to form a small structure. Small structure. This is called as primitive streak. So what happens here? This primitive streak, slowly it will elongate towards the cranial end and it will form a bulge towards the cranial end. Next, what happens here? Again in this pear shaped structure, this primitive streak will slowly form an elevation or bulge at the cranial part. This bulge is called as pharyngeal node or Hensen's node. What is this called? Pharyngeal node or Hensen's node. Now this is all surrounded by what cells? Epiblast cells. So this is how your primitive streak is formed. All these are epiblast cells. Next, what happens here next? All these cells of the epiblast, they will slowly invaginate inside the primitive streak. Okay, all these cells, they will slowly invaginate inside the primitive streak. So before this, a small invagination is formed at the cranial end of the primitive streak. So from this invagination, all the cells of the hypoblast, they will come and invaginate. Now, at the structure, let's see how actually in this cross section, how we will see this structure. We will see this structure. This is your epiblast. This is your invagination of primitive streak, epiblast, and this is your hypoblast. Understood? So this is your epiblast, this is your hypoblast, and this is your primitive streak, all surrounded by what cells? Epiblast cells. So what we have told, slowly these epiblast cells are invaginating. So same here, these epiblast cells will invaginate, and slowly what they will do, they will replace the cells of the hypoblast into endoderm. So they will replace the cells into what cells? Endoderm. So that's how your lowermost or outermost layer called as ectoderm is formed. Now, the remaining cells, they will occupy the space that is present between the epiblast and the ectoderm. So this center layer is called as what? Mesoderm. Now the remaining cells that are present in the epiblast, they will change into what cells? Ectoderm. So this is how the three layers, ectoderm, mesoderm and endoderm are formed. Ectoderm, mesoderm and endoderm are formed.